Hi, I'm Dr. Loreen Allphin, professor at Brigham Young University, and today I'm going to present some research that was done with the U.S. Forest Service at the Desert Experimental Range on the Frisco Clover. Trifolium friscanum, or the Frisco Clover, is known from the San Francisco, the Wawa, the Beaver Lake, and the Tunnel Springs Mountains in Beaver and Millard counties of western Utah. For this study, we focused our efforts on the Tunnel Springs Range located within the Desert Experimental Range. And you can see in these photos pictures of the Tunnel Springs and the highest peak in the left photo. Over the past few years, the Utah Division of Wildlife Resources has been considering reintroducing bighorn sheep into the Tunnel Springs Range. Given the continued drought in this area and talks about bighorn sheep reintroduction, the U.S. Forest Service was concerned about the status of the Frisco Clover on Forest Service lands. We initiated this study in conjunction with the U.S. Forest Service with three main objectives in mind. The first was to determine the extent and distribution of populations of the Frisco Clover within the Desert Experimental Range. The second was to demographically monitor the Frisco Clover populations to better determine trends. And the third was to monitor any additional natural or anthropogenic factors that might be affecting conservation of the Frisco Clover, especially in light of the possibility of reintroduction of desert bighorn sheep. For our demographic monitoring, we set up a 25-meter long-term monitoring transect through the largest and the most extensive of the populations of the Frisco Clover in the DER. This transect was set up in 2020. At the time, 100 tagged plants were tagged using the point center quarter method, and at each sampling point, a Daubenmeier frame was placed to ascertain the community structure or the associated plant community at each site. Individual plants that were tagged were assessed for several vegetative and reproductive characteristics. We also made note of any herbivory or any disturbances to individual plants or to the site. We returned to this long-term transect in 2021 and reread all of the vegetative characteristics for all tagged plants, and we also determined mortality rates of tagged individuals. An additional year of monitoring is planned for 2022. In this table, you can see our reproductive and vegetative measurements that were taken in 2020 and 2021. As you can see, our tagged plants grew over the time interval, as you might expect. They had significantly more rosettes per plant, longer leaves per individual, the plants were taller, plant height, and the percentage of plants with inflorescences also increased. However, the most important thing that I would like to highlight from these data is the low reproductive success. Not only was there a low percentage of plants that actually flowered, but there were very few flower stalks per individual. So in 2020, only 3.3 on average, or an average of 3 flowers per individual, inflorescences per individual. And in 2021, about 1 inflorescence per individual. In fact, in 2021, the number of flowering stalks per individual only being 1, we were unable to collect any fruits and didn't feel it ethical to collect any fruits to assess seed to ovule ratios. So I'll only address the seed to ovule ratios and the fruit to flower that were ascertained in 2020. And we found fruit to flower ratios only about 41% of the fruits developed from the flowers, and the seed to ovule ratios only about 14 to 15% of the ovules developed into viable seeds. It's an extremely low seed set for this species. Now, as you might expect from a clover, we saw evidence of herbivore utilization. So if you can see here, of our tagged individuals in 2020 and our tagged individuals in 2021, about 45% in 2020 were utilized, showing some level of being eaten. And in 2021, it was about 43%. If you count those that weren't utilized, the overall mean utilization per plant was about 9% of the plant utilized, or about 5%. Now, when you looked at utilization of just those that showed utilization, what was the mean amount of utilization if they were utilized? We found that about 15% of the plant on average was eaten in 2020, and about 12% of individual plants were eaten. The individual plants themselves were eaten utilized in 2021. And if you notice this plant on the left, you can see a mat of Trifolium friscanum there. And if you notice in that picture, there are little pellets, little round pellets, okay, very small, looking like maybe they belong to a lagomorph. So we thought maybe they belonged to a cottontail of some sort. At the long-term monitoring site, though, we also did see fecal pellets associated with deer. And so we collected those so that we might be able to ascertain what those species were that could potentially be utilizing the Frisco clover. So from those fecal samples, we performed what's called fecal DNA analysis. And from the fecal samples of the lagomorph and the deer that we collected, we wanted to determine if it was actually the species that we thought it was, what the sample belonged to. And also we wanted to see if we could reconstruct the diet of the herbivore and determine how much of the diet was made up of the Frisco clover. Indeed, the species that we picked up that I thought might be a lagomorph in the cottontail was indeed Sylovagus natalii, or the mountain cottontail. And the deer samples did belong to the mule deer. We did pick up Trifolium friscanum in 2 of the 3 cottontail DNA samples. So 2 of the 3 fecal samples contained Trifolium friscanum. However, it was in very low concentrations in those samples and made up only between 2 and 5% of the diet of that individual cottontail from which the sample came. Moreover, we did not find Trifolium in any of the deer samples we collected. So it appears that the main culprit for herbivory at these sites appears to be the mountain cottontail. And it does appear to eat, but it doesn't make up a significant amount of the total quantity of food that they eat. As you recall, we also set out Dobermeyer frames along our long-term transect to establish the associated plant community. We set out Dobermeyer frames at 25 sampling points along our long-term transect, and the results from 2020 and 2021 are given in this table. As you can see, the sites are very rocky. Bare rock accounts for between 55 and 60% of the cover in 2020 and 2021 at these sites. In addition, what we found is that there was a high percentage of the ground that was covered by cryptobiotic soil crusts. So when soil was there was a large percentage of cryptobiotic soil crust, making up between 15 and 18.5% of the soil crust. In fact, the soil crusts appear to be increasing. So between 15 in 2020 and 18.5% in 2021. Also note that 88% in 2021 of the frames we placed had cryptobiotic soil crust in it, so they appear to be increasing. The total vegetative associated species in this, the total vegetative cover, also appeared to decrease over the time interval. So from 2020 to 2021, most of the species on here appeared to decrease in abundance, with the exception of the Trifolium friscanum, which increased in cover from 2020 to 21 and increased in frequency from 20 to 21. In addition, most of the abundant associated species were the Limas ambiguous grass, Stenotus acaulis, Castanea scabrida, and Cercocarpus intricatus. But all of these species either remained about the same in abundance or decreased in abundance over the time interval from 20 to 21. So less total vegetative cover at all the sites over the time interval. Now, as you remember, one of our objectives, in addition to the long-term monitoring, was to determine the distribution and extent of populations of Frisco clover on DER lands. In order to do this, we surveyed the entire Tunnel Springs range in the DER and determined population number and size of population and any threats to any of the populations at every site where we found them throughout the Tunnel Springs range. And so the results of our data are given on this table here. The long-term site was indeed the largest, between 200 and 250 individuals, which we tagged over 100. And you can see that the long-term site, you can see the elevation, the GPS locality, and the estimated individuals. So there were, in addition to the long-term sites, 7 subpopulations that we found on the range. Most of these were very small. in size. The largest had about between 60 and 65 individuals. And but there's one thing I want to point out here that was evident, is that the percentage of plants that we counted at these sites that had inflorescences. So remember, our long term, we had less than 35% of the plants that were flowering or had inflorescences. Notice here, site 2 had about 17 to 18%, about 18% with inflorescences. Notice that sites 3 and site 4 had no plants flowering, and the highest was about 20% flowering, but most of the sites had very low percentage of the plants flowering. And we didn't see evidence of herbivory of lagomorphs like we did at a long-term site, but the plants seemed to be decadent in parts and drying out. There appeared to be evidence that these sites were dry and that drought may be playing into the few number of inflorescences that we found at this site. So once again, we found 7 subpopulations in addition to the long-term located throughout, but they were very small populations and have really low reproductive success at these sites. So in summary, the research that we did for the past 2 years in 2020 and 2021 at the DER showed that very few plants have inflorescences on them at all sites. In addition, Trifolium friscanum, if they did have inflorescences, there were few per plant. The highest mean in 2020 was 3.3 and only about 1 in 2021. The annual mortality rate that we found between 2020 and 2021 was about 4.6 at our long term. In addition, we found no recruitment at our long-term site. We only found 2 small plants across all survey sites. So we found even at our survey sites the lack of a presence of seedlings. Seedlings are extremely rare and recruitment appears to be rare. The mats are growing, so reproduction appears to be primarily vegetative. We also saw much dieback, which I hadn't talked about 28% of the plants were dying back. So thus, we do not recommend that the division introduce bighorn sheep to the desert experimental range at this time, because it's a highly palatable species. The lagomorphs are eating it, and it already has low reproductive success. So at this time, until we see that these populations are doing well and reproducing, we do not recommend it. Really quickly before we finish, I do want to mention that we visited a BLM site for the Frisco Clover on the northwest side of the Tunnel Springs. It's a lower elevation site. We didn't do studies on this because we were funded through the U.S. Forest Service. However, we did visit and wanted to ascertain what was happening at this site. Originally, Dwayne Atwood, several years ago, estimated 2,000 individuals of Trifolium at the site. We found nowhere near that number. We counted and scoured and only found 300 individuals. There's no way there could be more than 400 at the site. And of the 300 that we saw, we only found 3 of those that had any inflorescences on them. We saw no fruit development, and we saw the site appeared to be heavily trampled and eaten by domestic sheep, as evidenced by many tracks and domestic sheep feces at the site. And at the site, we didn't see cryptobiotic soil crusts. They seemed to be heavily disturbed, which is further worries for this really fragile species. And as we know, the species warranted listing, and we recommend that more studies be done on this species, including those populations on BLM land, to really ascertain how this species is doing because of the drought and everything else that has been happening at these sites. It's time that we look at the species in greater detail. Any questions?